All right, Jess, we can begin. Take it from here.
Okay, sounds great. I'll go ahead and share screen. And just double checking that you're seeing a full screen of slides and not slides with notes. Yep, okay. All right. Well, thanks so much, first of all, Vivek, Lindsay, for having me here to speak for the MDOT community. Happy Halloween to everyone. I was debating coming in costume, but I figured maybe the background would would help us to celebrate. I'm thrilled to be able to present the work that we've been doing on the Digital Biomarker Discovery Project, which is an open source toolbox for biosignal analysis. And my hope is that this can be sort of an interactive seminar where if you have any questions, please feel free to unmute and ask questions. The goal is a lot of the tools that we are developing, we want the community to be able to leverage and also contribute to. So if there are any questions, anything is unclear, or you have an idea for something that could make this better, please feel free to either unmute and hop in or send me an email. So my Twitter is up here, but my email is jessielyn.dunn@duke.edu. So that would also work to get in contact. So the motivation for the DBDP is the idea that we have kind of these ever expanding options for biosignal measurement. So a lot of us here in this audience work with more traditional consumer smartwatches, maybe even research grade devices that we purchase from research companies or that we develop in-house. But what we're seeing is that this is not just a research or clinical trend. This is something that is, continuing as smart clothing and as sort of wellness and fitness and clinical merged together in this space, that there are many different devices and many different wearables that are generating tons of information that we could be leveraging toward healthcare. And so that's what we're doing with the development of digital biomarkers. And so I'll talk a little bit about what I mean when I say digital biomarkers in a moment. But before I dive into that, I want to just mention some of the challenges in this area. So while we can envision a lot of the applications of all of these new types of measurement tools, we have challenges on the data handling side. So issues with how much we can trust the data coming off of these sensors, issues with how we can understand how that data was generated and how algorithms that use that data come to conclusions. We have issues with interoperability and standards, trying to make sure that when we're talking about a term, we mean the same thing as somebody else, so that we're not talking past each other in these communities. There's also an issue of huge amounts of data coming in and trying to understand how to deal with those large amounts of data. And so hopefully if there's time in this talk, we'll get to some of the biosignal compression toolboxes that we've been developing on this front to better understand how we can compress data in a meaningful way toward digital biomarker development. And then an issue that we've been seeing across not just digital health, but all areas of healthcare and research that involve large-scale data and the development of algorithms. We have issues with how data is shared and keeping data private, or protecting it in a way that protects the people who donate that data, as well as handling bias and inequities. So understanding what are the inherent problems in the system that will affect the technologies that we develop. So unfortunately, this is not a 4-hour talk, so I can't address all of these issues. So what I'm going to be tackling in the realm of the DBDP today will be this sensor accuracy and validation challenge and the data deluge. So the talk today will include an introduction. I'll talk a little bit more about my lab, the work that we're doing, and what I mean when I say digital biomarkers. And then we'll address the challenge of accuracy. And there will be 2 case studies that I'll present toward this. And there will be a collab, so an online notebook that has code that if you are more technical or if you're interested in following along, you can actually do this hands-on approach as I go through the talk. There's also an option not to touch that and to just listen to the talk. And then I'll talk a little bit about some data compression work and where we hope to go next. So my lab is the Big Ideas Lab. We are the biomedical informatics group, and what we do is we integrate data engineering and analytics. So Big Ideas actually stands for something here. And what this translates to in practice is that we are trying to develop and test tools and infrastructure using biomedical and health data for early detection, intervention, and prevention of disease. This is a really broad goal, and I think most people who are in this webinar probably also share similar goals. And so again, this is some of the motivation for the work that we're doing, is that we can come together as a community to accelerate our progress in this space. So when I'm talking about digital biomarkers, here's what I mean. I'm talking about digitally collected data, so we could consider any continuous stream of data. Maybe that's location from a smartphone GPS, maybe that's continuous measurements of heart rate every minute over the course of days, months, years, and we transform those streams of data into an indicator of a health outcome. So that could be an indicator of infection or prediabetes or a more traditional biomarker like blood glucose. So there are a variety of ways that we could think to design a digital biomarker. But the basic structure of this is that we have some target outcomes. So these might be clinical measurements, which could be more traditional biomarkers. This could be survey data. So perhaps somebody's reporting in symptoms that they may be having, or this could be illness events, for example, taken from an electronic health record. And what we're doing is we're developing prediction algorithms for these outcomes based on different digital health measurements. So again, these can be anything that is measured from a sensor that is either on body or ambient in the environment that's giving us information about a person's physiology, behaviors, and so on. And so with that, we have particular applications that we're working toward, but you can imagine that this framework can be applied to a variety of different situations. So here are just a few of the areas that we're working in. We have been doing a lot with infection detection. We have a study called Cove-Identify, where we have been using digital biomarkers from consumer smartwatches like Fitbit, Apple, Garmin, to indicate whether or not somebody is likely to have a COVID infection. Prior to that, we were working with flu, rhinovirus, RSV, and making a lot of progress on that front. We've been working in the cardiometabolic disease space and chronic diseases to understand over very long trajectories, how can we tell if somebody is improving or developing more problems associated with a disease. And we are expanding out into mental health and women's health. And you can imagine that if there's an area that you work in, the likelihood is that there is digital data that's relevant to that area. So what we've seen with these various kinds of digital biomarkers that we're developing, that there's a common structure to the development. And with that, it led us to develop this digital biomarker discovery pipeline. And so essentially what this is, it's a repository of code, data, and algorithms that make discovering digital biomarkers more accessible and also help to establish best practices for the field. And there are many individuals and entities that are working in the space of establishing best practices. So one of the areas that we're working on is accumulating what's already being done in the community, so that if somebody is wanting to start the journey of developing a digital biomarker, they have the information about what data standards look like, about how to validate that a device is measuring what they hope it's measuring. And so really what this talk is centered on today is what are some of the current capabilities of the DBDP, and where do we hope to go next, and how do we hope that you'll join us in that journey? So if you want to check out what is available, you can check out dbdp.org. And what you'll see on there is starting with the modules that we have available. So a lot of what we have worked on and what we find that a lot of collaborators spend time on is once you actually get data from a wearable device, there's a lot of work that goes into cleaning up that data. So sometimes we refer to this as sort of the data janitorial work. There's just a lot of hands-on processing that needs to happen. And a lot of this is systematic. It happens the same way for any sort of data set, and some of that is specific to the data types. So there's a lot of information about pre-processing data, exploratory data analysis, so how to visualize your data that can help you to iterate with that pre-processing method. And then depending on the types of data that you have coming in, we have different modules to, for example, go from a blood volume pulse from a PPG sensor to measures of heart rate variability or resting heart rate. One of the things that I really want to emphasize here is that while we are putting up the modules that we develop as a part of the research in my lab and with our collaborators at Duke and elsewhere, lots of individuals are developing many different modules that could be stitched together to accelerate digital biomarker discovery. So if this is of interest, we are working with a broad variety of researchers to add modules. And in fact, we've worked with several groups from MDOT. We've worked with Santosh Kumar, with Loki Nataranjanan to get modules that have been developed by others up on the DBDP. And so hopefully you might find either something that's missing that you would like to see, and please contact us because we're trying to work to make more modules for the community, or you may have something that you think could benefit the community or other coders might be able to build on and make even better. So with that comes the need for extensive documentation. And so with any open source software community, we need detailed instructions for how to contribute code, for how to monitor any issues that come up if you try to run somebody else's code, or vice versa, somebody tries to run your code. And so there's a lot of iterative work that goes on here to make sure that what we are doing is building on the shoulders of giants. And as a part of that, one of the benefits to submitting your own code is that you have people that are helping to find bugs and potentially find solutions for those bugs as well. And vice versa, if you're using somebody else's code, you can get a lot of new ideas about how to do analysis. So one of the other things that's really useful about this open source community is access to data. So it's not only useful to be able to develop code to make digital biomarkers, but oftentimes, we're starting a project, we have some idea, but we don't necessarily have the resources to collect primary data. And so we've been developing this digital health data repository, which you can imagine is akin to other types of data repositories, but is very specific to digital health data, meaning data coming from wearable or ambient sensors that tells us something about physiology or behavior. So if you're ever looking for a data set, either if you're teaching classes or taking classes or trying a new research project, there's data here to be shared. And if you have a data set that you think would benefit by sharing and perhaps having lots of eyes on the data and building off of maybe previous work that you've already done, there's an opportunity also to deposit data here. So the last piece of DBDP that I want to mention is the educational component that we've been working on, which has educational materials for a broad variety of interest levels. So we have tutorials, and we'll talk through some of those today with the case studies and the collab notebooks. We also have blog posts, which are useful for people who are just getting started in this field, or if there's a niche area of digital biomarker development that either we or partners with this work have had experience with and have been able to write a blog post to share that experience. So again, if this is sort of relevant to you, if you have been hitting your head against the wall, trying to develop some project, and you finally overcome some really challenging roadblocks, I would really encourage you to submit a blog about what that process was, so that we as researchers aren't continuously reinventing the wheel. And then finally, our goal is to do some community and K-12 outreach. This is also working on the bias and equity front of the digital biomarker discovery pipeline. And part of this is to get people into this pipeline early, so they know that this field exists and they can begin contributing and doing research and doing reading and trying out collab notebooks from an early stage. And as a researcher, you might think, well, a lot of this work is very complicated. It seems too complicated to explain to a room of high school students. Actually, we have found that not to be the case at all. We have found, students who are in their teens know when they put on a watch that data is coming off of that, and to actually see them working on that data in real time is super inspiring. And students are a lot more tech savvy, I think, than we sometimes give them credit for. So there's a lot of work going on in that space that we're also hoping to expand on. And with that, we've been collaborating with OpenM Health and with IDA-SIM to try to stitch together the various aspects of digital biomarker discovery. So what this slide is intended to be is sort of a map, so that you can find where you are and how that fits in with this architecture. So if you are completely new to digital biomarkers and you have no idea where to start, DBDP Ed is a great place to start with the blog posts, the tutorials, and maybe moving on to some of the contributor guides and understanding how this community is working together. Perhaps you have wearables and mobile devices and you're hoping to collect data, or you're planning a study and you don't know what that data is going to look like or how you're going to get from the device to data. That's where OpenM Health comes in helping to establish what are those data structures and what can you expect. Perhaps you don't have any data to work with. Well, that's where the DHDR, the Digital Health Data Repository, comes into play, because you can start to brainstorm ideas by pairing the data in the DHDR with some of the modules in the DBDP. And then finally, maybe you already have data from wearables and mobile health devices, and you're wanting to build off of existing code or perhaps contribute new digital biomarkers. That's where these DBDP modules come in. And in an ideal world, we are, as a community, developing and validating each other's digital biomarkers to make them such that they can be used by the broader community and really be trustworthy. So with that, I'll take a quick pause in case there are any questions about what is the DBDP, what is it that we're trying to accomplish, how is it that, in the various aspects of digital health, your work might fit into the framework?
This is Mani from UCLA. So I had a quick question. Is there an overarching software architecture in which the tools that one contributes plug in the sense, are there APIs and all, or is it I guess that's my question. Like, how do things interact with each other?
Yeah, so this is in some ways an early days implementation of this. Right now, if you go to dbdp.org, you'll see that the way that these code modules are stored are as GitHub repos. We have these repos have code in usually Python or R. And a lot of times they have they take an input from another module as, an output from a previous module as an input to another module. So if you are code savvy, you could envision stitching these modules together with a wrapper script. We understand that that is not accessible for everyone. And so on the other side of this work, we are developing a GUI-based system where it's actually drag and drop. And you can drop these modules together and stitch them together so that the output from one becomes the input of the other. And for those who are familiar with bioinformatics tools, this is kind of akin to Galaxy, where we have a bunch of bioinformatics tools that have been developed sort of sequentially by a community, and the outputs of one become inputs to the other. And the vision for this is that this continues to evolve such that, you know, we do have maybe one environment where all of this is living. But for the meantime, in order to make it possible for everyone to be contributing basic Python, R, MATLAB, Julia, you name it, basically whatever your language is, that's how we're starting out, and really welcoming any insight into where this could go.
Okay, thanks.
Hi, this is Amir Rahmani from UCI. A follow-up question. So is it then supposed to be something like that programmers or users would run locally on their machines, or there will be like backend support or servers to kind of, I just want to understand the model. Yeah.
Right. So that is the vision, again, is that this would be something that would be run in a cloud environment. So, as we were getting started, Vivek and I were talking about kind of where is DBDP, how could it connect with MDOT and other efforts in this space. We have projects that are local to us and our collaborators that we are running in the cloud. Right now, we don't have the resources, we don't have the funding to be able to open that up to other investigators. But that is the hope as to where we're going to be able to make that possible. And whether that is that DBDP creates that environment, or that DBDP stitches into an existing digital health cloud environment that has analytics, for example, the MD2K machines, that could be an option. So right now, if you were to log on and use this, the expectation is you're bringing your own environment. But the goal is that that's the first implementation.
Thank you.
I see a hand up from Blaine.
Hi, yeah. So my question is, in this graph you say wearables and mobile health devices. Is this platform designed to start once you have data, or are there tools for data collection from either consumer or custom wearable devices?
That's a great question. And that's why, we've been working so closely with OpenM Health, is because we want to make it accessible from any part of the pipeline. So essentially, you may have wearables, you may not have wearables, and there are resources to get you set up. So with OpenM Health, there are these tools called shims, and the shims enable you to pull data from different devices. Not every device is represented here. So, Fitbit, Apple Watch, the more common consumer devices are represented. But again, the goal is that to move away from this sort of one-off development that we as researchers have been doing, which is to say, I want to pull data from Fitbit using the Fitbit API. In-house, I develop that code, and now nobody else has access to it. So it makes a lot more sense for us to not be sort of spending efforts doing that over and over again, but rather putting that up for others to build on, and then building a better tool. So we have basics in place. And so again, I really do want to emphasize that this is the skeleton of, in the theme of Halloween, this is the skeleton of the infrastructure that we hope is going to flourish with all of us working together on it. So, we just have some starting points, and we hope to just continue to build and grow this resource. So with that for time, I'll keep going just to demonstrate what this actually looks like in practice. So going back to these challenges that we talked about, one of the major challenges is sensor accuracy and validation. And when we're talking about sensor in the case of DBDP that we'll talk about today, we're talking about sensors that exist in consumer accessible devices. You could imagine extending this to any device that you might be developing in-house, but for the purposes of today, we're going to just focus on devices that you could purchase as a consumer. So we have two case studies that we'll talk through. The first one is going to be the validation of heart rate extracted from consumer wearables against a reference standard ECG device. So we'll talk through what would be the motivating question for wanting to do that. The second case study that we'll talk through is the assessment and monitoring of a consumer wearable for a specific application in circadian rhythm monitoring, and showing how there are gaps in existing tools that we sometimes only find by actually running a study. And then once we discover those gaps, we can develop patches that can then be used for future studies. So I'll start with case study number 1. And the idea here is that you own a smartwatch and you are using it during your physical activity and activities of daily living. You want to know how well that watch measures your heart rate compared to a gold standard ECG. So this is a real case study that we actually published on. So if you wanted to read what the outcome of this sort of work would be, you can actually see the publication. And so that's in NPJ Digital Medicine. It's a 2020 paper that was published by my former PhD student, Dr. Brene Bent. And so what we did as a part of this study was we collected data on 53 people that were between the ages of 18 and 54, and we had representation from a variety of skin tone types using the Fitzpatrick scale. And the data collection protocol consisted of these 5 tasks. So we had somebody sit at rest at baseline, do a paced deep breathing, do physical activity on a treadmill. So this was just a treadmill walk, do a seated rest, and a typing task. So that was to get the wrist movement. And we looked across 4 different consumer grade devices that sort of any person who is not necessarily a researcher would use. And we also looked at some more research oriented devices, the Empatica and the BioVotion. And we compared the heart rate measurements during these different tasks and across these different skin tone and body profiles to the ground truth, what we call the ground truth here, ECG. And one of the things to note is that the data that we are getting from these consumer devices is using PPG, or photo applied tomography. So one of the reasons that we're so interested in the effect of skin tone is that we know that this optical method of measuring heart rate can have interference from melanin in the skin, which gives skin pigmentation. And so one of the things that we wanted to know was, is there an effect of skin tone on the accuracy of these heart rate measurements? So what we're comparing here is the optical-based PPG measurements from these different devices to the electrical-based measurements of ECG. And you can see briefly kind of what these signals might look like. And what we typically use to gather the beat-to-beat information is these R-to-R intervals. So from the ECG or from the PPG, we're looking from peak to peak to gather information about the heart rate. So what this process looks like, it seems like, we have a straightforward question. We want to know, how accurate are these devices compared to some ground truth? Well, there's a lot of computational processing steps that need to go on in order to be able to answer that question. So first, we need to be able to access that data and pre-process the data. We have different sorts of data. So we have ECG data that we need to pre-process and extract these beat-to-beat intervals from. We want to resample and merge the data with the PPG data and perform alignment of that data. And then finally, visualize and compare. So there are multiple ways that this could be done. I want to mention some of that work that we're doing with the graphical user interface, because the next thing that I'm going to show you after that is going to be a Colab notebook with code. And so to have an idea of kind of these 2 different ways of potentially performing this process, we can see that, and I hope that this video works, but the idea here is we're going to import that data into the GUI, and we're going to see how you can actually interact with that data without having to do any sort of coding. Then we'll move to the coding version. So what's happening here is, and again, this is hot off the presses, this is a brand new tool that we're working on. We upload our sample data and we're flipping through that data to just see what it looks like. What are our columns? What we might want to do is, if we find that there are missing values, we may want to do some sort of imputation. So we have the option to do that, to fill missing values using this completely graphical user interface. So you can see we have backfill, forward fill, interpolate, and all of a sudden now a process which would have had to be coded previously can now be done via point and click. One of the challenges here is that we want to make sure that people really know their data well. So there's a lot of steps that would go into sort of pre-processing that we would hope to see happen before or after this process. But what you could then do is move to data analysis once you perform all of that pre-processing. So you can put in the data from these different devices. You can look at a single individual in the study. You could look at multiple individuals in the study. And you can essentially take a look at the differences between different devices. So let's say we want to compare the Garmin with the Empatica and see how similar are those measurements. We could perform a regression and calculate an R-squared. We can look at different values of these measurements. And there's a variety of different ways that this can be done. We can create these correlation matrices to understand how similar or different devices are. And so all of this essentially is being done in a GUI, in a graphical user interface. What is underlying that is the code for this case study. So again, if you're interested and you're wanting to follow along what's going on in these slides, please feel free to pull up bit.ly/DBDP case study 2. And essentially you can follow these instructions and you can essentially just point and click in this Colab notebook to actually see what these steps are become processed with the underlying code. So again, the idea here is that we would use this method to continue to build upon this code. So perhaps you're going through the case study and you see that, I would love to see a visualization like X, Y, and Z. Well, this is all on GitHub. You could fork one of our repos, you could create a branch, you could do that and you could push it back. And all of a sudden your idea becomes a part of this case study. So that's really the idea here. Okay, so what we're doing then is in this Colab notebook, we're performing ECG pre-processing. So I will just quickly hop out of these slides just to show you what you should be seeing on your Colab screen if this is something that you are interested in doing. So hopefully you all can see a Colab notebook. Okay, great. So essentially what you can do if you want to follow along is just hit these play buttons. You can hit run anyway. Hyung is my PhD student. I trust her. And what you can do is you can sort of see as we run this code what actually is happening in real time. But what I'll be showing you is just the results of this. Okay. Are there any questions about that? Is anybody having trouble getting to this Colab notebook that we can quickly troubleshoot? Okay. Okay, so if you run through that notebook, what you'll see is that we have an ECG data preprocessing that consists of these steps here. And ultimately, we're going from our raw ECG signal that came off of the ECG patch that we used for this study to the R peak detection. And you can see each of these steps and how the data changes along each of these steps. Finally, what you'll see if you run all of these steps in the Colab notebook is that you can perform comparison of the different devices to each other. So previously we saw that in the GUI, but here what you're doing is running code that you can actually change and alter to perform different sorts of analyses. So here what we're looking at is the comparison within a particular individual, looking at the Apple Watch versus the Garmin looking at the Garmin versus the ECG, the Empatica versus the Fitbit versus the ECG. So these are just some examples, but you could imagine doing, you know, the Xiaomi Mi Band versus the Fitbit. You could imagine kind of expanding out the way you're doing this analysis. And so that code is there for you to play with and just get a feel for what that would look like. And now in this code, we're looking within a single individual. So we're looking at the error of these devices. And remember, going back to that original question we were asking is, which of these devices is, you know, maybe the most accurate for me to use during a fitness episode, during exercise. And so we could look at the mean directional errors within this individual and note that some people wear watches differently than others. Some people tighten it, some people wear it loosely, some people wear it higher or lower on the arm. And so you may be interested in looking for your own data, which device is most accurate. And then you may also be interested in looking across a population. So looking at different types of devices, whether that's consumer versus research grade, whether that's during certain types of activities or across different skin tones. How well do these devices perform based on their mean average error or mean directional error? So all of this data and all of this code is accessible for you to run. The key takeaways from this study, and again, you can read more about this if you're interested on the paper, is that the device type affects heart rate accuracy. So the different devices have different sort of levels of trustworthiness of data depending on what is the context of the measurement. During physical activity across nearly all devices, we saw that the accuracy of the heart rate decreased dramatically. And on average, it was a 30% decrease in accuracy. So this is quite important if you're running a physical activity study where you want to see how are people's heart rates changing during different levels of intensity of exercise. Knowing that these devices have decreased accuracy is super important. So this might be a notebook that you could use to collect new data from new devices before you run a study to figure out how accurate are these devices compared to the ECG. And we were pleased to see that there was no change in accuracy, given that there had been a lot of popular media reports that this was potentially an issue. We did not see this in the devices that we used or the data that we used. But again, you can take this into your own hands and try it yourself. So why do we care about that? Why do we care about the accuracy of heart rate? Well, you know, we can imagine different settings in which these devices may be used. So we have this sort of overall wellness setting. Maybe you're tracking your resting heart rate on a daily basis. Maybe one measurement in accuracy is not that big of a deal. But when we start to move to a fitness setting where you're monitoring your heart rate during a high intensity activity, and maybe you're the type of person who has some sort of cardiovascular abnormality, a single measurement may actually really matter. In a clinical setting, it definitely matters. So depending on the setting, we might have to tweak which devices we're using and how we're interpreting that data. And then we can also ask questions about what level of data do we need? Is a single measurement of heart rate enough? Do we want to see the PPG waveforms? Do we need interbeat interval data? So there's a lot that we can start to ask in digging into this Colab notebook. And one of the things that, you know, we built on from that previous study was that ability to do the COVID-19 detection using these consumer smartwatches. So again, this is getting to this idea that the ability of this research to build to the next level depends on these building blocks really being strong and in place. And the more that we work together to build these, the more that we can expand out and build on each other's work. With that, I may skip going into the details of the next case study because I do want to make sure that we have time for questions, but I will give the information for this next case study and just a quick overview of what it is so you could try it on your own if you'd like to. So this will be the same link, except this one is now DBDP case study 2, or sorry, case study 1. We had reversed the order, so 1 and 2 are swapped, but you should be able to find both. And again, you can click through that Colab notebook and see how this works on your own. This study, the idea behind it is that we are tracking circadian rhythms you are having a tough time sleeping, you may be sleeping at odd times. We know that this is related to many different mental health conditions. We have a data set that was collected by our collaborators, and the publication is in the Journal of Biological Rhythms. All of this information is in that collab notebook. Ultimately, what you will end up doing through that notebook is looking at the sleep detection algorithm, which is these blue squares here, and finding that when people are sleeping at odd hours, their sleep doesn't get captured. So ultimately, what is happening is with this notebook, you'll be imputing where sleep would have occurred. So we're taking an algorithm that used to be based on clock time for sleep and converting it into being a data-informed algorithm for sleep detection. So feel free to go through that on your own, and if you have any questions, don't hesitate to reach out. So finally, with that, I do want to mention this issue of the data deluge, because this is an issue from both an implementation standpoint of handling these large volumes of data, as well as a practical standpoint of where do we go from here? There was a question about, are we hosting this in the cloud so that anybody can come and run this code? The answer is no, partially because of the volumes of data. As the volumes become larger and larger, this becomes a more expensive prospect. And so we want to know on multiple different fronts, how can we address that? And so in thinking through this data deluge issue, we know that we have huge amounts of data that are coming online every day, and that there are lots of trade-offs between having a sufficient resolution of data, meaning how frequently we collect a measurement, and how useful that data is and how much that addresses sort of issues with the battery life of the device or the ability to store that data for a prolonged period of time. And one of the questions that we've been asking is, how can we minimize data volumes while maximizing the accuracy of the digital biomarkers that we're developing? So there are a few ways we could think to do this, and you may have other ideas as well. We could reduce the amount of signal level or raw data that we collect at the initial experiment. We can develop some application-driven data compression methods that preserve the integrity of that digital health data for digital biomarker development, or we can intelligently aggregate data together in a way that reduces the overall data volume, but makes it still useful for our purposes. So going back to the heart rate example, if we're thinking about data coming from these different devices, we might have our ECG signal that is a super high frequency, like 1,000 hertz, 1,000 measurements per second. We might have various levels of PPG data that we could collect at a high resolution, like 64 hertz, relatively high, or a low sampling rate, so maybe 16 hertz. And the question may become, well, if our goal is to be able to recapitulate heart rate, what level of signal frequency do we need to collect in order to achieve that goal? And how much data do we need to store in order to be able to achieve that goal in the future? And so as a part of this study, we did a downsampling analysis where we looked at actually, if we cut the frequency of the data by half each time, where are we no longer able to recapitulate information about heart rate, and specifically here, heart rate variability? And what we found was that we could reduce the sampling frequency by more than half. And the red asterisks here are showing where we start to have statistical differences in sort of the distributions of the data. But up until 21 hertz, from 64 to 21 hertz, we're really not dramatically changing the ability to get these heart rate variability measurements. So this then begs the question of, well, can we be more intuitive in the way that we decide what sampling rates to use? The beauty of this is that we not only reduce the data storage. So if we look at data storage required for 24 hours worth of data, when we cut our sampling rate in half, we cut our storage in half, we cut our costs in half. So on the practical side, this really does matter. When you're thinking about reading in data, we cut the time to read in the data in half, we cut the memory usage in half. There's really a lot of benefit to this. The last piece that I will mention as a part of this is that we have a lot of different sensors that we have signals coming in from. And so what we might want to do is also understand not only what should be the resolution that we collect data at, but if we want to compress the signals of data coming in from these different sensors, can we actually leverage the structure of those signals in order to make more intelligent decisions about compression methods? So as a part of this study, what we were looking at was balancing this sort of trade-off between how much we can compress data, so that's this compression ratio here, and how much the data integrity remains the same. So how similar is that compressed data to the original data, which is this percent root mean squared difference. So we want to minimize this difference and we want to maximize this compression ratio. And in this case, we used a set of pretty standard data compression methods, and we paired them with very standard encoding methods and essentially tried different combinations of these to see which ones worked best for which signals. And ultimately, this became a data compression pipeline that is now available on the DBDP. So if you have any signals of these kinds and you want to see, am I able to reduce my storage footprint, or am I able to reduce my memory usage, you could apply this data compression pipeline to that data, and it's available today for you to download that code. As it turns out from this particular study, we found the optimal compression methods for ECG and PPG that took advantage of the cyclical nature of the data. For EDA, electrodermal activity, and accelerometry that took advantage of sort of peakiness of data, and skin temperature that took advantage of the consistency in the values of data. So the hope is, again, that we continue to learn from each other as we are developing these new methods to say, what are best practices? How did this work for me? And can somebody else then use it to accelerate their research? So I'll go ahead and wrap up this talk. We talked through in detail some case studies related to accuracy, related to dealing with large amounts of data. And ultimately, our goals here are that we want to be able to leverage wearables and sensor data to provide patient empowerment, precision therapies, just-in-time interventions, and improved access to care. The only way that we can really make this vision come true is if we all work together. So this is, again, a plug for getting involved, or if you have ideas on how we can bring existing communities together, you can contribute code, you could write a case study on research you've already done. You can just simply learn what is out there. Let's make this happen. For anybody who might be attending, there's an AMIA conference coming up. We'll be talking through some more details about DBDP and going through some more case studies. And I could not possibly have done any of this without my group and our excellent collaborators, particularly Mobashir Shanli, who is on the faculty market, for those who may know of open positions, Haiyang Jiang, a PhD student in my group, and my former PhD student, Brene Bent, who have put together all of these excellent resources. So I will stop sharing, and if there are any questions, I'm glad to answer them.
Excellent. First, thank you for a wonderful presentation. It's always gratifying to see a mHealth scholar come in with this. And there are several here, and I see some hands going up, so I'll open it up. Amir?
Sure. First of all, thank you so much. It was a very great talk, and I believe this initiative would be very useful to bring folks together. I have one question, because sometimes some of these studies, for example, the validation study, it's not only signal dependent, but also the final kind of outcome you want to measure. For instance, if you want to validate PPG for pulse ox, that's a different thing, right? For example, and we know that the skin tone has a kind of impact on on pulses, but not for HR or HRV because they're so peak dependent, and the peaks are quite resilient to, for example, the signal. So is there a way that we can, for example, like also specify the target, or I assume because it's open, I think anyone should be able to do that, but I just wanted to see if that's it's part of the plan.
Thanks, Amir, for bringing this up. That's a great point and a great question, because a lot of this is domain driven, and that, different people have different related questions, but the ultimate goal can be different. Even if we're talking about heart rate variability, that can mean various things. We could be looking at an SDNN metric or an RMSSD metric. So, all of these have relationships, but also differences. And so part of that is this idea with being able to sort of fork repositories or build kind of these branches off of the repositories to say, I'm going to work with the heart rate variability validation module, but my interest is in particularly using the Empatica watch, and particularly using this in patients who have tachycardia. Right. So all of a sudden we have these very specific case studies, but then somebody else may come in who's also studying patients with tachycardia and wouldn't have looked at RMSSD of those patients because they just don't have time to develop that module. But because it already exists, they could leverage that. And so the answer to your question is the domain matters a lot, and the documentation matters a lot. So that's part of what my team here is doing, is sort of curating the code deposition. So if somebody deposits code, we have sort of a list of questions. And if you've ever deposited code, for example, or data to PhysioNet, you've probably seen something similar, that there's a lot of questions about what is the use case? Who else could benefit from this? Explain, if there's data involved, be very detailed about that, comment the code. And then we have people in our group who actually run the code and make sure that it's intuitive. But the more that people annotate their own work, the more likely it is to be used and cited. So there's also kind of that benefit that we try to add in. Does that answer your question?
Yes, thank you. That's what I also expected. Just wanted to make sure. Yeah, thank you so much.
Okay, thanks. I see Nabil's hand up.
Yeah, thank you so much, Jessalyn. This is very interesting and much needed work. I actually have quite a few of my students and staff here, because I think this is something that's so critical and so important. And so I wanted to ask you, you touched on this a little bit. So a few questions regarding quality control. And you mentioned, that you have some folks that also run some of the code prior to sort of providing it to the public. Another question, with these, and I guess everyone sort of has their own unique case study, but, with ECG, for example, if we wanted to, let's say, insert a, noise detection module within the pre-processing phase, how, I see the code's public, but how simple, or how, what are some of the challenges that we might face working with your system? And I guess my last question has to do with and I see you giving this talk as one example of continuing this effort, but how, because I've seen a lot of this start, right? Like the wearable development kit that was published in IMWIT, or you become 3 years ago. Bullying started this, a decade or two ago as well with activity recognition. How are you going to sustain this and your plans there? Would love to hear a little bit about that. Sorry for all the questions, maybe.
No, thank you. These are both great questions, and both things that I would really like to talk about. To your first question of how to actually use this. Again, I want to emphasize that this is the early days of this. So the hope is that people who are interested come together. I mentioned already that, we've been working with IDASIM, with Santosh Kumar's group, with a lot of the people who have been in this space for a while. So not just trying to kind of build our own thing, but really have the community work together to build this and stitch pieces of this together. So, it doesn't matter what it's called, as long as it's serving the function that it is bringing the community together to build on work that's already been done. So currently, the way that that looks is, the home is GitHub. There are code modules there. And if you wanted to create a branch of code or add in a new module, all of that will live within the GitHub environment. The hope is that this continues to evolve to the point where we actually, have a cloud environment where the modules are maybe a little bit more seamless in how they work together. But GitHub is actually, in some ways, it doesn't totally make sense to reinvent the wheel on how to collaborate on code, because GitHub is actually pretty good for that. So as much as we can leverage the existing infrastructure, that also, it seems to be working well. And again, this is an evolving process. And if you have ideas, if you want to get involved, again, I can't emphasize that enough. Please email me. We have a contact us form on the website. Like it really is something that we want the community to work together on. To your second point about, what is the sustainability of this? So all of this started, and I'm actually remiss that I didn't mention that this was initially supported by a grant from the Chan Zuckerberg Initiative for open source software. And we have been doing a lot of work on understanding how open source software environments and communities evolve. So, we've trained alongside people who developed Matplotlib and NumPy and sort of like you name the open source environment and how does that work? And really what it is, it's the community members who are developing code want to see it used, right? We don't want to develop these one-off projects that die. And so it does come down to, us sort of getting together and making these choices to make our code public, to stitch it together, to put in that slightly extra effort to do that. And so we are starting sort of like a listserv, and we will have sort of a quarterly meeting for subgroups. And the first step is to get to find out who wants to be a part of it. And then once we go from there, there will be more sort of boots on the ground work to be involved. Definitely trainees are, hugely welcome to be a part of that as well.
Thank you, Jason.
Question from Tushar.
Hey, Professor. I am glad to attend this presentation. My research exactly, kind of tackles the problems you were mentioning in the presentation, and would love to collaborate, contribute to your co-profile, the open source project that you are developing. I had two quick questions about the content in the presentation. I know there's just 1 minute left. One was about this accuracy of sensors compared to the gold ECG standard. So that figure it kind of indicated that Apple Watch is doing the best, I guess, in terms of variability from gold ECG. So I was just hoping, what was the definition of heart rate that you use for that? Like whether it's instantaneous heart rate or is it averaged over a window? Because, from Apple Watch, we usually cannot get very finely resolution heart readings of ECG, and so what was the definition of HR used to make that figure? And I had a second question real quick about the compression comparison that you showed. I was trying to search your page for a paper. Is there a paper for that published already? Could you send a link for that?
Yes. So very briefly, the different devices for that study had different resolutions of sampling. And so the heart rate measurements varied based on the device. So that's all explained in detail in that paper. And I will go ahead and send sort of the references in the chat here to those papers. And there is a paper for the data compression pipeline. I actually don't have that reference handy, but perhaps, Vivek, is there a way that we could maybe add information for attendees in some other way?
Yes, we can.
Okay.
The presentation will be curated and on our YouTube channel, and we'll put the links inside of that.
Okay, so I'll go ahead and add details for the compression pipeline as well.
Thank you, Professor.
We have.
I'm not sure. Do we have time for one more
We'll
question?
take one more. Yingji, you wanted to ask a question?
Yeah, thank you so much, Professor Jasmine and Duham. It's a great presentation, and also thanks so much for building this platform, not only for the community who work on health-related issues, but also for researchers like me who from ecology and environmental science and policy. Because we are currently working on looking at the connection between nature exposure and human health. I have background in ecology, so the nature side is pretty clear to me, but we have very little knowledge in the health side. So I look at the webpage and a lot of modules, especially the module related to wearables, especially very helpful for us to explore. And in addition to this, I do have one question, specific question related to the wearables. You mentioned several health-related indicators already included in the module of pipelines. I was wondering, have you considered the GPS, like the geolocation of each individuals or the wearables? For example, if we can track the trajectory of the movement of the people, we can link with satellite images and get to know how often people connect with nature, like green space and other exposures. Thanks.
Thanks. This is a great question. It actually leads to some of the work that we're interested in privacy and security, because you can imagine of all of the types of digital health data that we might collect, GPS is a very sensitive type of data. And so in thinking, what does it mean to de-identify GPS data, for example? I don't think we totally have clarity on that yet. And if we want to make code public, I think it's difficult to do that and not actually have any practice data to work with. And so then we would kind of be needing sort of an open GPS data set. For those of you who have seen some of the stories about how the US military bases were revealed by some Strava running data being made public, I mean, there are a lot of challenges that we often don't see until sort of we have hindsight. So I would say of the data types that we've been working with, we have not really delved into GPS. If there is somebody who is an expert in GPS and would like to take that on, that is a whole branch that needs to be worked on and really could be improved, again, with all of these questions about sort of privacy and ethics in mind. And I think we're over time, so I will hand it back to Vivek now. Thanks so much.